We are dealing with the chapter of MSc second SAM, botany. Polytrichum is here, Polytrichum life cycle is here, which we are dealing with. Occurrence, Polytrichum has a worldwide distribution. They are common in cold temperature and tropical regions in shaded places. General structure, rhizome, it is the horizontal part and underground. It bears 3 rows of small brown leaves and small rhizoids. It is quite different from marchantials and anthracoidals as well. The upright leafy shoot, these are longer and they are developed from the rhizome, which contains spiral leaves. The leaves, these are with broad bases. The upper part is green, whereas the lower part is brown. Green cells are lamely and photosynthetic part. These have the thick midrib and the margins are 1 cell thick. 1 or 2 layers of squamous tissues above the epidermis and the thin-walled parenchymatous tissues in the center. The upper part is photosynthetic. You see lamely and the gaps, which is characterized by lamely. The stem, TS of the stem shows 3 parts: epidermis, medulla and cortex. The medulla is again differentiated into the central zone and peripheral zone. The cortex consists of thick-walled cells, and the innermost layer is called mantle with a starch grains. The starch grains are therein. Epidermis is found over the cortex, which is thin layered. The leaf is differentiated, well developed, advanced as compared to those of marchantials and jungle mammals, other anthracoidals as well, where such leaves are not characterized and these are absent. The air part is too advanced and developed by bearing medulla and cortex. Now, the life cycle. The life cycle, it takes place by means of protonema and fragmentation, by means of vegetative reproduction. So it is the kind of vegetative reproduction that is differentiated from other orders. Protonema, the spores germinate to form protonema and protonemal buds, which give rise to the geometrophytic plant. The rhizome gives rise to the leaves and aerial part. These are separated and form the independent plant body. Sexual reproduction, it takes place by means of antheridia and archegonia, as usual in all bryophytes. Antheridium, these are clustered, longer and surrounded by paraphyses and perigonial leaves. The antheridium is developed through the antheridial initial cell and antheridial mother cells, and the anthrazoids are ultimately formed. So if you see details in the antheridial development, and you can look into all other developmental stages inside the antheridial development. So it is the life cycle. Yeah, antheridia are stalked and longer. The matured coiled bioflagellate anthrazoids are liberated through the operculum. Bioflagellate anthrazoids. So we'll be looking into the part of life cycle. Archegonium, these are clustered and longer as well, and they are surrounded by the pericytial leaves. as you see in Markensia. Leaves maybe 3 to 6, paragytial leaves maybe 3 to 6 in a cluster. The archegonium is developed from the initial cell to the central cell, and the central cell cuts off neck cells and the neck canal cells and the venter. This venter develops egg and the neck canal cells develop a neck. The neck is longer, 8 to 9, 8 to 12. Neck canal cells may be there inside the neck. The mature archegonium contains cover cells on the top that opens for the anthrazoids at the time of maturity. The longer neck and a broader venter with an egg. So see, on maturity of the antheridium and archegonium, the process of fertilization is going to take place. Fertilization. The sex organs on maturity come in contact by releasing anthrazoids and opening the part of neck. The anthrazoids are coming out of the antheridium. They swim in water and they are ready to fertilize the egg. Some archegonium opens with an organic fluid. It is sugar-like organic fluid which chemotactically attracts anthrazoids. Bifilous anthrazoids are swimming into the water. They are, however, come in contact with the archegonium neck, where only one anthrazoid fuses with the egg. After fertilization, the zygote will be formed. It usually happens in all bryophytes. What is essential, and the fertilization takes place inside the archegonium, inside the venter. Zygote is formed, and this zygote gives rise to the sporophyte later on. The sporophyte develops through the zygote, 2x, and is advanced type. It has an outer and an inner air spaces. When it is developed in the form of the capsule, the sporophyte is very advanced and more advanced than other Markensials and Anthocerators younger manuals. The sporophyte is specific in air spaces, and a long columella is formed. And the two laterally arranged spore sacs, these are also very specific. They are not found in sphagnum. They are not found in Markensia and Palea, Anthoceros, etc. It is surrounded by the calyptra. It is divided into seta, apophysis, and capsule. The endothecium forms a spores and operculum. Calyptra covers the whole sporogonium. You see here. The peristomial teeth play a very important role, significant role in case of descents of the capsule on maturity. So the role of peristomial teeth is very specific here to detach the capsule. Elatas are not here. Elatas and electrophores are not found here, and therefore they do not take part in descents. So it is quite different from Markensia and Palea. Dispersal of the spores from the possibly opened peristomial teeth takes place, and dispersal by wind takes place. The spores germinate with the exosporium and endosporium under the favorable conditions. The spores give rise to the protonema and buds, and they are developed in the form of a male and female gametophyte. So the protonema is very specific, distinguished structure here, which has been developed in case of Polytrichum. The protonema develops buds and give rise to the male gametophyte and female gametophyte separately. The sporophyte is dependent on the gametophyte, and thus the alternation of generation is characterized. The capsule of Polytrichum is curved and hairy to differentiate from the other kinds of capsular structures and sporophytic structures as well. So you see the life cycle here. Which is differentiated from the stage of gametophyte and sporophytic structure. That's all. Thank you.